We are now part. Okay, so we are. This work was mostly done in Andalusian Center for Regenerative Medicine. Now we have transferred to Principal Felipe Research Center, and we are part of the National Stem Cell Bank. So, as you have heard from the previous talk, so induced pluripotent stem cells are the stem cells obtained from somatic cells by introducing 4 to 5 defined transcription factors. So after a reprogramming period, these cells become fully pluripotent, and they can. The two basic characteristics of these cells are their capacity to divide indefinitely in cell culture, and also to differentiate into any mature cell type of the human body. These two characteristics are also characteristics of embryonic stem cells, which are derived from human embryos, from the blastocytes. However, there is a problem with the use of human embryonic stem cells due to the ethical issues and also the immunogenicity problems. So the value actually of the iPS cells is that we can derive iPS cells from patients, and we can then differentiate these iPS cells into the cells targeted by disease. Therefore, we are getting access to the disease-targeted cells, which we otherwise don't have access to. In that way, we can create disease models in which we can study the disease pathogenesis, and the same platform can serve to screen for drugs for their efficacy and toxicity. We can screen the small molecules and also gene therapy. And also, in the case of genetic diseases, we can identify the causative genes. Also, if we introduce a healthy copy of the gene in the case of genetic diseases, we can put these cells back into the patient. So these cells can serve as a cell therapeutic source. So we are interested in hereditary retinal dystrophies. So you know that the retina is a multicellular organ, so it has several cellular layers. However, the two cell types affected in hereditary retinal dystrophies are photoreceptors and retinal pigment epithelial cells. So the photoreceptors are the light-sensing cells, and the RPE cells are responsible for the homeostasis in the retina. So in the case of hereditary retinal dystrophies, the generation begins in one cell type and leads to the secondary generation in another cell type, because these two cell types are interacting, and which cell type the generation begins depends on the causative gene. So when we plan to make a disease model, so we need to know, or we should know where our gene is acting, whether in RPE cells and photoreceptor. So for most, this will give us the information toward which cell type we need to differentiate our iPS cells. So for most of the genes involved in hereditary retinal dystrophy, we know where they are acting, whether at the level of RPE or photoreceptors, but in some cases, such as, for example, the splicing factors which induce retinitis pigmentosa, we still don't know whether they are responsible, they cause first RPE degeneration or photoreceptor degeneration. So the stages of when we want to make a disease model is that we need to obtain somatic cells from healthy patients, and also from patients having a retinal dystrophy. So we expand our somatic cells, we reprogram their somatic cells into the iPS cells. So in this case, we are going to obtain normal iPS cells, and in this case, we are going to obtain iPS cells which still bear the genetic defect. So after a differentiation process, we differentiate these iPS cells into normal RPE, normal photoreceptors. However, if we differentiate iPS cells bearing a genetic defect, we are getting RPE cells and photoreceptors with a genetic mutation. However, the disease phenotype is going to be expressed only in one of these two cell types, depending on which gene is causing the degenerative disease. So we are collaborating with Dr. Carmen Ayuso in Fundación Jiménez Díaz in Madrid, and we obtained several biopsies from several patients affected with retinitis pigmentosa. And one of the patients that we got had a mutation, was genotyped with the mutations in the MERTK gene. So this gene codes for a MER receptor tyrosine kinase, and we know that this tyrosine kinase is involved in the process of phagocytosis. So the process of phagocytosis is a daily process performed by RPE cells. So RPE cells phagocytose outer segments of the photoreceptors, and this helps to renew the membranes of the photoreceptors, which are damaged due to the constant light exposure. And what this MERTK protein does is it's a signaling molecule. So when the outer segments are bound to the receptor, then the FACT kinase is activated and activates the MERTK protein, which signals inside the RPE cells and enables the phagocytosis to occur. So MERTK deficiency is known in animal models, in spontaneous animal models, rats and MERTK northbound mice, and also several families with the MERTK disease have been described. So a common thing between these two, the animal models and human disease, is that there is always an accumulation of the debris in the subretinal phase, and this is due to the defective phagocytosis. So knowing that we have a patient with a MERTK mutation, so our expectation is that if we take patients' fibroblasts and convert them into the RPE cells, these RPE cells are going to be defective in the phagocytosis. So, as I mentioned, we got a biopsy from the retinitis pigmentosa patient. This is a healthy control, and this is patients from which we took the biopsies. So the patient was genotyped with a mutation. This is a novel mutation in MERTK. It hasn't been described so far. So it's a serine 331 frameshift, which causes the premature stop signal. So the full protein is 99 amino acids longer protein. However, the predicted truncated protein in this patient is only 334 amino acids. So you can see that this protein is lacking this truncated protein is lacking the kinase domain here, which is actually the effector domain, which transmits the signal into the cell interior. So presumably this patient will not have the kinase effect. And also these truncated proteins are usually degraded in the cell, so they are not even visible in the cell. So we took biopsies, as I said, and we generated patient-specific RPE cells in this way. So we first cultured the primary cultures of patients' fibroblasts. So at this level, there is no difference, morphological differences between the fibroblasts. Then we generated iPS cells from the healthy patient and MERTK, a patient bearing the MERTK mutation. So this is a typical iPS colony. These cells grow as the colonies, and this around what you can see are feeder cells, because these cells are cultured in vitro on feeder cells. At this level, also, there is no morphological differences between the healthy and affected iPS cells. And you then differentiated these cells into the RPE cells. So these are typical RPE cells with polygonal morphology and pigmentation. And also at this level, there is no morphological differences between the healthy patient and the healthy individual and patient's RPE cells. We tested the presence of RPE-specific markers. So we wanted to be sure whether our RPE cells that we are obtaining in vitro are really mature RPE cells. So we tested, for example, in the healthy individual RPE monolayer, we tested for the presence of these specific characteristic RPE markers, such as MITF-01, PralBP-BEST1, sodium potassium ATPase. So these are all known to be specific RPE markers. When we did the same assay with the affected individual, we didn't observe any differences. So we also observed the expression of these RPE-specific markers. However, when we looked at the protein expression of MERTK protein, we see a nice expression of the MERTK protein in the healthy individual RPEs. So we can also see that this merticase is a focal section. This is a basolateral section, and you can see here this is a vertical section. So you can see here that merticase is correctly distributed on the apical part of the RPE monolayer. So you can see here that this is monolayer. The blue is nuclei of the RPE cells. However, when we looked at the expression of the merticase protein in the RPE monolayers obtained from the patient, we absolutely don't see any merticase expression. So this is also a vertical simulation on focal microscopy, and there is absolutely no merticase expression in the case of the patient. So this proves us that we are able to obtain in vitro the mature RPE cells, and also this result confirms that we are able to obtain mature RPE cells which are patient-specific. We also confirmed the absence of merticase in by Western blot. You can see that these are two monolayers derived from the patients loaded on a Western blot, and there is an absence of the bands that correspond. There is absence of the bands that correspond to merticase in the case of the patient. However, in the healthy individual, these merticase bands are present there. So, knowing that merticase is responsible for phagocytosis by RPE cells, so we performed a phagocytosis assay in vitro. So this assay is performed in the way that you incubate the RPE cells cultured in a dish. You incubate with outer segments which are labeled with the fluorescence, and the outer segments we usually use are derived from bovine or pig retina. So in the case of the healthy patient, we are able to observe that these cells, the RPE monolayer derived from the healthy individual, is absolutely capable of phagocytosis. So this is a confocal microscopy in the basolateral section. You can see that the fluorescence is present inside the cells. So this is a vertical section, so you can see that the fluorescence is really inside the cells. However, when we do the same assay on the RPE monolayers derived from the patients, we can see that these monolayers are absolutely not capable of phagocytosis. So there is absolutely nothing, no fluorescence inside these cells. We also only see a small amount of photoreceptor outer segments present at the area of microvilli. So again, this confirms that we are able to get fully mature and also functional RPE cells, and that these RPE cells are really patient-specific. So this confirms that this is a proof of concept that our strategy is working. So knowing that we are able to get patient-specific cells, now we can do all kinds of studies with this model. So next, what we did is we added to the patient's RPE monolayers, we added a wild type copy of the wild type merticase gene through the adenovirus vector, and in the monolayers we didn't express any level of merticase. When we add this vector after several days, we are able to see the expression to a certain extent of the merticase protein. So in case we confirm that these layers are functional also in the phagocytosis assay, we can consider this approach for a cell therapeutic approach. So to conclude, okay, this is the last one. So using IPS technology, we obtain patient-specific RPE cells which can reproduce the retinitis pigmentosa phenotype in vitro. So in our case, we had patients with the merticase mutation, so we knew which phenotype to expect, and this is what we confirmed. But the value of this strategy is that we can use this approach in future for diseases in RD patients with completely unknown disease mechanism. So, for example, retinitis pigmentosa caused by splicing factor mutations, we don't know the disease mechanism at all in these cases. So by observing the phenotype, we would go back and we would study the disease mechanism. Carmen Ayuso in Madrid, and these are other collaborators which have participated in this work.